seminar, and as such, it's a work in progress seminar. So I'm going to take this time to
This
show
is Junior.
Yes.
Junior?
Yeah.
Not muted.
So I'm going to take some time today to introduce you to some cutting edge research coming from a CIEHS lab. So I'm going to start by introducing some concepts, and then get into our emerging pilot study and show you the very beginnings of our data. What I'm hoping from you at the end of this is to get some thoughts, some feedback, and potentially some collaboration to push this project forward even more. So the title of my talk is looking at heavy metal roles in brain aging, and in particular, I'll be focusing on chromium neurotoxicity and chromium as a gerontogen. I'll introduce some of those topics as we go. I've broken up my talk into 3 sections. The first is looking at aging and environmental pollution and how these are 2 sides of a toxic coin. I'm then going to take that toxic aging coin and bring it to the brain, give you a brief introduction of the brain and how the brain ages and how chemicals can contribute to that. And then I'll dive into our pilot study, looking at how or if chromium prematurely ages the brain. To start, I want to take some time to talk about aging and talk about environmental pollution and how these are interacting. The first thing I hope you can appreciate from this figure is we are entering an aging epidemic. The top figure shows you the percentage of the population over the age of 65 for 2015, and the colors here show you the percentage of the population that's over that age 65. And then the bottom figure shows you 2050, what's projected for that, and what you can see there is most of the world will have reached a point where at least 25% of the population is over the age of 65. On the heads side of this coin, we're looking at how environmental pollution impacts aged individuals. In the last 100 years or so, we've seen a global average age increase from 30 to 70. And then it's projected that within the next 30 years, at least 800,000 Americans will see their 100th birthday. So we have a situation where we have people living really long periods of time as an aged individual. And this is all coinciding with a global increase in environmental pollution, which really started about 100 years ago with our Industrial Revolution. So that begs the question of how do these environmental pollutants affect aged people? We know from gerontology research that an aged body has different biology from a young body or from a developing body. But we don't yet understand how these chemicals might affect an aged body differently from a younger body. So the key research question is, are older adults more susceptible to environmental pollutants? And there's some indication that yes, they are. On the flip side of this coin, we have environmental pollution aging the young. So this is a concept where chemicals can induce or accelerate aging phenotypes, and these are known as gerontogens. There's a few that have been studied already and are considered gerontogens, but there's a lot more that still need to be studied. And I'm proposing the metals as a key chemical for that, for gerontogenicity. So the key research question here is, are environmental pollutants prematurely aging key organ systems? So to introduce you quickly to gerontogen, it's defined as a chemical or an agent that accelerates or exacerbates the natural process of aging. The Free Dictionary provides a slightly different definition, and from the Wikipedia page, you can see it's very brief. It doesn't go into a whole lot of detail. We don't know a whole lot about them. And that's largely because this is a term that was only coined back in 1987 by George Martin. And since then, according, as you can see from PubMed, only 2 papers have been published that mention the word gerontogen. So now I want to bring this idea of this toxic aging coin and dive into the brain. And in particular, I'm going to focus on the tails side of this coin, looking at how chemicals may induce brain aging or accelerate brain aging. First, I want to give you a quick introduction to the brain. It's a very complex organ, as you can see in the figure on the left, with a whole bunch of different regions pointed out. All these different regions have different compositions of cells, different compositions of elements, and they all have different functions in the brain. The figure on the right shows you the cerebral vasculature, which is a part of the brain that's often disregarded. In the last 10 or 15 years, there's been a spike in interest in this neurovascular structure. Classically, when we think of the brain, we think of neurons, and neurons are postmitotic. So anything that might affect the cell cycle is disregarded for brain research. However, we now firmly understand that there are 8 or 9 different cell types in the brain, and I'm going to introduce you to a few of them. So first we have our neuron in the middle there, which is an electrical active cell that's responsible for our cognition and our thinking and our information processing. I might use a pointer for this slide real quick. So there's our neuron, and then after our neuron, I want to introduce you to the oligodendrocyte, which is responsible for putting these myelin sheaths on the axons. You can think of this as sort of the rubber sheathing around electrical wiring, so it makes the signaling go smoother and faster. And then we have our microglia, which is kind of like a resident macrophage. So normally these are dormant. They have small cell bodies and large processes, and they're kind of surveying the brain parenchyma. And then when they receive a inflammatory signal, they become activated, they retract those processes, they develop a more robust morphology, kind of like what you see here, and then they move to the site of action where they typically phagocytose stuff and degrade it, just like a macrophage does. We have astrocytes, which are kind of the caretakers of the brain. On the one hand, they are supporting neurons, providing nutrients, clearing up synapses after neurotransmission. And on the other hand, they are helping to maintain the blood-brain barrier, which I'll get into on the next slide. And we have our ependymal cells, which are found in a few spots of the brain and are responsible for creating cerebrospinal fluid. Now for the cerebral vasculature, there's this new concept called the Neurovascular Unit, which is a composition of a variety of cells as well. First, we have our astrocytic end feet, as you saw from the previous slide. Underneath those, we have pericytes that are wrapping themselves around the vasculature. They are important for signaling between the brain and the blood, and they're important for maintaining the blood-brain barrier, which is a tight junction structure that keeps stuff out of the brain. And then within the vasculature, you have the endothelial cell, which makes the cell wall, which makes the blood vessel walls. And so any one of these cells can be vulnerable to neurotoxic insults. So next, I want to talk a bit about what aging looks like in the brain. Some general observations: we see loss of dendritic spines. You see decreased oligodendrocyte number. Both of these are going to reduce the efficiency of brain signaling. We see a decreased brain volumes in multiple regions with sex differences occurring, but we don't see any total neuron loss. Rather, it seems to be the neurons are shrinking, at least for primates. And there's overall changes in gene expression, again, with sex differences. And we also see with age an increase in risk for developing neurodegenerative diseases. Typically, it's considered after the age of 65, your risk of developing one of these diseases goes up significantly. I want to focus for a moment on Alzheimer's disease, because this is the most common neurodegenerative disease and is often viewed as accelerated brain aging. So to briefly introduce you to Alzheimer's, the figure on the left, I want you to understand that this is a progressive neurodegenerative disease. The blue on the brain shows you what parts of the brain are affected at different stages, with it mostly starting in the prefrontal cortex and then spreading throughout the cortex and into the hippocampus. These are the regions that are most important for executive function and learning and memory. On the figure on the top right, you can get a sense of what a healthy brain looks like versus an end-stage Alzheimer's disease brain, and you can clearly see that brain atrophy. There's two key proteins to understand. One is amyloid beta, which is normally found in the plasma membrane, and due to improper processing, it breaks apart and aggregates, forming these amyloid plaques within the cells. And then there's tau, which is normally found in the axons of neurons and are important for maintaining that axonal structure. And those also, due to improper processing and hyperphosphorylation, they tend to aggregate and form these neurofibrillary tangles outside of the neurons. We also see effects on the neurovascular unit, both in aging and in Alzheimer's disease. The first thing that is important to note is that we have a loss of pericytes, and because of that loss of pericytes, the neurovascular unit becomes leaky, and that leakiness allows serum proteins to enter the brain and red blood cells and circulating monocytes. So this creates a toxic environment to neurons and a pro-inflammatory environment. It induces neuronal injury. It induces microglial activation and astrocyte activation. And so this is there's a developing hypothesis that this is occurring prior to amyloid beta aggregation or neurofibrillary tangle formation, and may be one of the earliest events occurring in Alzheimer's disease and aging. Altogether, there have been 9 hallmarks of brain aging identified as depicted here. The key thing I want you to think about with me with regards to these is how chemicals might induce these, and thereby how chemicals might accelerate the aging process in younger individuals. So moving forward with this tail side of the coin of how chemicals accelerate brain aging, we asked the question of, are there brain gerontogens? As I alluded to earlier, there's 4 that have been characterized that are considered your classic gerontogens. Those are cigarette smoke, PM2.5, ozone, and arsenic. And overall, what we see with these are progressive neurodegeneration, neuroinflammation occurring in a lot of parts of the brain, increased aggregation of amyloid beta and phosphorylated tau. And behaviorally, we see impaired executive function, impaired learning and memory ability, and a loss of speed of learning and memory. So these are, you can think of these as kind of like the hallmarks that we're looking for with gerontogens. Now I'm proposing metals as a gerontogen, and we have well established that metals are neurotoxic. Classically, we recognize lead and mercury, arsenic and manganese as neurotoxicants. Mercury, of course, being associated with the Mad Hatter. Lead, of course, being associated with loss of cognition, and in fact, the higher blood level you have, the lower your IQ, or the more your IQ falls. And there's a whole bunch of learning and memory impairments that occur in children with high blood levels. And we have these newer metals that are now being looked at as neurotoxic, such as cadmium, which you're most likely to be exposed to either through cigarette smoke or through polluted soil that gets into our food. And hexavalent chromium, as I'll get into a little bit later, which is certainly neurotoxic. It's just a question of how and perhaps where. Another thing I want you to appreciate about metals in the brain is they tend to accumulate in different areas, and as such, they have different behavioral effects. Manganese, which can accumulate in the basal ganglia and the striatum, kind of in the middle of the brain here. This can induce a disease similar to Parkinson's disease. It's called manganism. Mostly happens with welders who are occupationally exposed to manganese. Lead, as I mentioned, is associated with loss of ability to learn, and that's because it accumulates in the hippocampus. And iron has been shown to accumulate in the substantia nigra, which is the region of the brain most susceptible to neurodegeneration in Parkinson's disease. And so there's a whole lot of research looking at how iron might be a contributor to Parkinson's disease development. So to start answering the question of whether or not metals are brain gerontogens, so far there's been one paper that has considered this. This is a paper from Iqbal and Ahmed from 2019. And here they used a unique group of animal models where they had two groups of young animals, one given saline, one given a combination of 40% high-fat diet and metals in their drinking water for just over a month. And then an elderly mouse group that was only given saline. And what they observed was these brain aging phenotypes in these high-fat diet and metal-exposed group that looked very similar to what they observed in the elderly mice. Things such as decreased acetylcholinesterase, which impairs cholinergic signaling, decreased neuron counts in the hippocampus and the cortex, increased oxidative stress, and behaviorally they observed impaired learning and memory and heightened anxiety. Again, much similar to what they observed in the elderly mice. So, whether or not metals are brain gerontogens has yet to be thoroughly studied. What I'm proposing to focus on is how metals affect genomic instability and cellular senescence in the brain. And with these two topics in mind, I've built up this research hypothesis. One, considering the fact that metals are well known to induce genomic instability. They are well documented to induce cellular senescence and inflammation. We observe DNA damage and chromosome instability early in neurodegenerative diseases, both in humans and in animal models. So my overall hypothesis is that metals induce aging in the brain that is mediated by persistent DNA damage and results in cellular senescence and this senescence-associated secretory phenotype, or SASP, expression. The SASP is essentially 56 different cytokines that are being released when the cells shift to this senescent state. So to depict this graphically, you've got DNA damage accumulation occurring normally in life, and I'm proposing that metals such as chromium are contributing to that. You have aging processes that occur at the molecular level that induce cellular senescence, which is essentially an aged cell. Those then express SASP, which kind of triggers other cells to express SASP and induces this systemic inflammation, which we observe in an aged body. So now I'd like to dive into the final section of my talk, looking at our pilot study and whether or not chromium might prematurely age the brain. You might ask, why chromium? Well, for me, there's 4 key reasons. One, we have widespread exposure, both globally and locally here in Louisville. There's a significant health need with key data gaps. It targets key aging hallmarks, and we had samples that were readily available here at U of L. So to come back to that widespread exposure, it is the 7th most abundant element in the Earth's crust. And within America, we release anywhere from 900 to 970 tons per year into the air. This is mostly coming from things such as burning fossil fuels like coal and oil, which Kentucky uses a lot, and also places like that produce cement, and it's found highly in the cement dust. And hexavalent chromium is common in Superfund sites, so it's a very big concern for environmental health. With regards to the health need and the data gaps, ATSDR considers it 17th on their hazard priority list. We have a limited understanding of chromium neurotoxicity. Most of the data has looked at reactive oxygen species and antioxidants. We don't know much about the mechanism behind chromium neurotoxicity, which is again something I'm trying to dive into myself. And currently there is no data on hexavalent chromium and brain aging. So anything that I come up with becomes a very new discovery and easily publishable. Chromium is known to induce various aging hallmarks. In particular, I'll be focusing on genomic instability. It's well established for that, though we don't know whether or not it induces any of that in the brain. And as I pointed out with my brain introduction, brain is traditionally looked at as post-mitotic, but we have a variety of cell types that continuously divide throughout life. And so we need to start asking whether or not there are genomic events going on in the brain in response to environmental chemicals. And chromium also induces cellular senescence in many cell types, but again, this is unknown in the brain. The samples I'm working with come from a collaboration between the WISE lab and the Kai lab. As you can see in the picture over here, there's myself and Chang Li from Kai lab. There's Haiyan, and behind me there's Edoya. This is from one of the days that we were collecting organs from our animals for this study. As I've mentioned, chromium is neurotoxic. We'll be publishing a review paper on this soon. It's both in humans and in animals. It's been shown to accumulate with age. The figure on the right there shows you where chromium accumulates in the human brain, with the highest accumulation occurring in the pituitary. And then the bottom is a rodent brain, either a mouse or a rat. And so far, there's only been one study that's done regional analysis looking at the pituitary and the hypothalamus. Otherwise, all the studies just report brain levels without giving any indication of what part of the brain they observed it. With animal models, we observe widespread neurodegeneration. So far, there's been 7 or 8 different species that have been looked at with chromium neurotoxicity. And then in addition to that widespread neurodegeneration, we see widespread oxidative damage, especially with lipid peroxidation, given how much lipid composition there is in the brain. And we see reduced expression of redox enzymes like glutathione and catalase. Behaviorally, both in humans and in animal models, we see social memory loss and spatial disorientation. And there's been 2 studies looking at children exposed to hexavalent chromium in the environment. One from Pennsylvania that looked at air chromium and found an association with childhood development of autism. And one study from Spain that looked at chromium in nearby rivers and found that the children at schools nearby those polluted rivers had developed impaired attention development. With regards to chromium as a gerontogen, there's no data on hexavalent chromium causing genotoxicity in the brain, but it's highly genotoxic in other organ systems. There is no data on chromium causing cellular senescence in the brain, but it does induce cellular senescence in other organ systems. So my hypothesis for this project is that hexavalent chromium induces persistent DNA damage in brain cells, which leads to the brain aging, again, with this DNA damage to cellular senescence to systemic inflammation paradigm. For this project, I'm working with 2 different models. One of them is a cell culture model, which are MO59J and K cells, and I'm looking to link genomic instability with senescence in brain cells. So far, I've only worked on objective 1 to determine some dose and time to try and induce toxicity in these cells. I've yet to start looking at cellular senescence in the cells. But after I figure that out, I'll move on to looking at clastogenic effects preceding or coinciding with senescence, and look to see if I can pharmacologically disrupt the DNA damage response and prevent cellular senescence. And for the rat model, I'm focused on trying to determine what regions of the brain are targeted by chromium. I've got a variety of data to share with you from this. Here, I'll be looking at genotoxic and cytotoxic endpoints in brain regions, looking at chromium deposition in a few regions. We're limited with how many we can look at. And then finally, I aim to look at inflammatory status in these regions that exhibit genotoxicity and cytotoxicity. So the first approach, where I'm working with the MO59J K cells, I'm aiming for proof of concept to show that chromium is genotoxic and can induce cellular senescence in brain cells. These cells were derived from a malignant glioblastoma of a 33-year-old male. And then the J cells naturally lost expression of their DNA protein kinase, which is an important protein in DNA damage repair pathway. And as such, the J cells are deficient in DNA damage break repair and are considered about 30 times more sensitive to ionizing radiation than their respective K cells. So far, I've just done a whole bunch of cytotoxicity analysis with these cells. On the left, I've got data from potassium dichromate, and on the right, for data from sodium chromate, using both the same hexavalent chromium concentrations, and on the x-axis, and the relative survival on the y-axis. And what's really intriguing here is with the potassium chromate, we see toxicity mostly occurring between 1 and 5 micromolar. But with the sodium chromate, we don't see a whole lot of cell death going on. In fact, it seems to benefit relative survival. So the future directions with this are to next look at chromium uptake in these cells, because that may account for some of the differences that we're seeing between potassium and sodium chromate. And we'll look at DNA damage assessment to look for genotoxicity in these brain cells, and chromosome instability to work on that paradigm that I'm proposing. And finally, look at protein analyses for cellular senescence. Next, I want to go into my animal model, where I'm trying to define what regions are targeted by hexavalent chromium. For our animal model, we are exposing rats to zinc chromate via oropharyngeal aspiration. So the rats are lightly sedated, and then their nose is pinched, and they're given zinc chromate with a pipette once a week for, so far we've done our 24-hour time point and our 90-day time point. 6 months, I think we're aiming to get done this year. And then we're doing 3 different doses of zinc chromate, both sexes. And like I mentioned, this is a collaboration between the WISE lab and the Chi lab, and we collect pretty much every major organ from these rats for various studies, as well as blood, urine, and tibia for biomarkers. For my endpoints, I'm looking at chromium accumulation, histology analyses, Comet assay, and protein and RNA quantification. And I'm looking at both the brain and the heart, because Lu Kai and I also have an interest in the heart-brain axis, but I'll have to save that topic for another presentation some other time. So to start, I'm looking for evidence of brain aging with immunohistochemistry. My goal here is to identify which brain regions and potentially which cell types are most vulnerable to hexavalent chromium genotoxicity and gerontogenicity. To probe this, I'm using silver stain to look at neurodegeneration. I've started to look at gamma H2X using IHC to detect DNA damage to detect DNA damage. And then after that, I'll be looking at cellular senescence with either beta-galactosidase or P16 INK4A, which are classic markers of cellular senescence. And we're looking at chromium accumulation in a couple of different regions. And eventually, I'm going to add antibodies to look at various cell types and figure out which ones are most vulnerable. So to start, I'm looking for evidence of neurodegeneration with silver stain. This is a classic neuroscience stain, which stains dying neurons this jet black color, and the rest of the tissue gets this kind of brown or amber color. I'm working with the whole brain, a whole brain hemisphere from one side of the brain that's fixed in formalin and paraffin embedded, using the Ph.D. Neurosilver kit and this Paxanos atlas of the rat brain to identify what regions exhibit this neurodegeneration. Now, the first one that was exciting to look for was the hippocampus, which you can see here in the control and here in the 0.4 treated zinc chromate. And right off the bat, what you can see is there's not a whole lot of jet black neurons. There's one here in the control, but in the 0.4 treated, we see a bunch more of them occurring. And to take a closer look at this, again, you see one here in the control, and you see about a dozen here in the 0.4 treated. So it's clear that neurodegeneration is happening, at least in the hippocampus. I also noticed it in the cerebellum, particularly the vermis of the cerebellum. But in the control, you can see there's a few. In the 0.4 treated, there's quite a bit more. And in the 0.8 treated, almost all of them are stained black. And a few other regions that I've identified are depicted here. Perhaps the most interesting so far is the thalamus, because that's been looked at for chromium accumulation in other studies. And to give you a sense of the cerebellum again, this part is the vermis, kind of like the inner channel of the cerebellum, whereas this part is the cerebellar cortex, and they have different functions. And in particular, I point that out because there's been studies with humans that show the vermis is more impacted by chromium and accumulates more chromium than the cerebellar cortex. Now, given that the WISE lab is proficient in common assay analyses with cell cultures, we thought we would take this on and look for DNA damage by common assay in our tissues. And especially because this had to be done with fresh tissue, this ended up being a few very busy days with a bunch of people being involved. For this, we microdissected the brain and the heart, brought those pieces of tissue through mechanical and enzymatic digestion, passed them through single gel electrophoresis and stained them with cyber green. And when we imaged them, they look kind of like this, where sort of the center of the nucleus is this bright white spot. And then through the gel electrophoresis, the DNA is brought across the gel and it creates this tail. So that's why it's called common assay, because the more DNA damage there is, the longer the tail you have. And we're focusing on two particular data points from this common assay. One is how long the tail is. That tells us how much the DNA is broken up. Smaller pieces are going to travel through the gel faster. And then on the flip side, we're looking at the intensity of the tail, which tells us how much DNA is in that tail. So here's the summary of the data from that. We've got results from the female on the left, results from the male on the right, corrected tail intensity at the top, and corrected tail length on the bottom. And right off the bat, it looks like there's not a whole lot going on. But what we can see is in the female cortex, there seems to be an increase in tail intensity and tail length, and also occurring in the female heart. But the males have some increase in tail intensity in a couple of the brain regions, and some in the heart. but don't seem to have a whole lot of effect going on with the tail length. Now, a couple of reasons why this might not look like a whole lot is one, we have a small n or a small sample size in our group, and two, as I mentioned, there are 8 or so cell types, and so if one cell type is affected, that might just get washed out with having all the other ones there. So next, I decided to go a little bit further with this and look at gamma H2X in the nuclei of various brain regions. This is what we would expect to see with a gamma H2X stain with untreated. There's some gamma H2X, but with the topocyte treated, which induces DNA double strand breaks, you can see there's quite a bit more gamma H2X being formed. So I'm doing 3 different endpoints for analyses. One is to look at ROI analysis, where we essentially trace the nucleus in the software, and it gives us a readout for DAPI and gamma H2X and how much signal is there. The next being foci analysis, where I count all the different gamma H2X foci within the nucleus. And finally, the number of gamma H2X positive cells per unit area, which I'm only just starting to get into, so I don't have any data to show, but I can show you what that looks like. So first, to look at the gamma H2X intensity. Here I've got an image of the cerebellum from a control animal and a couple nuclei from the cerebellum of a 0.8 treated animal. And this was just sort of a pilot experiment to see if it was working and to see if there's any immediate effect. So each of these was from one animal, and the number here indicates the number of cells I looked at from the cerebellum, the cortex, and the heart. And on the y-axis, we have the relative gamma H2X expression. These are all normalized to the average of the control to make the analysis a little bit easier to understand. And right off the bat, what you can see is both in the cerebellum and in the cortex, we have an increase in gamma H2X intensity with the highest dosed rats compared to the controls. But in the heart, we don't seem to have this effect. Next, I looked at gamma H2X foci. So here I've counted all these bright green dots within the nucleus. And on the y-axis, I have the average number of foci per nucleus. And again, what you can see is in both brain regions, we seem to have an increase in the number of foci per nucleus, but are lacking this effect in the heart. And then the next thing I'm adding into this is looking at the number of gamma H2X positive cells per region. So on the left, you can see a stitched image of the cortex from a control animal with a whole bunch of blue nuclei. And on the right, you can see from a cortex of a 0.8 treated animal, and there's a whole bunch of nuclei that are also brightly stained green. So I'll be quantifying that difference. We also wanted to ask where chromium is accumulating in the brain. Given that we have to microdissect the brain, we're limited with what regions we can look at, and we went for a few different sizes to try and figure out what regions we could. So we took the cortex, which is kind of up here, cerebellum sits back here, the hippocampus, which is kind of behind the tissue right here, and the hypothalamus down here, that was also a region that's been implicated and studied before. And then from the heart, we took the left ventricle, and we collected these samples and sent them to collaborators at University of New Mexico in Jim Liu's lab, where they did analysis for chromium, zinc, and iron. So first, I want to show you the data from our 24-hour time point, with the chromium results in the figure on the left and the zinc results on the right. Here, the females are in red and the males are in blue, and the regions on the x-axis. And what you can see with the females, we seem to have an increase in chromium in the hippocampus and perhaps in the cerebellum, whereas with the males, we have a much bigger increase in the hippocampus and not so much anywhere else after 24 hours. Perhaps a small dose-dependent increase in the heart. Whereas with the zinc levels, again with the females, looks like there are high levels in the hippocampus, but zinc chromate treatment doesn't increase that. And overall with the males, there doesn't seem to be an increase in zinc going on with our treatment. Now looking at the 90 days, what you can see is a slightly different pattern, where the males seem to have an accumulation of chromium in the hypothalamus, but nowhere else in the brain, and the females don't seem to have any accumulation going on in the brain. But we have this huge increase in chromium in the heart. I think when I quantified this, it was something like 12 to 17-fold difference of chromium in the heart of the treated animals compared to the control. But the zinc levels, we again see a different pattern. Overall, with the females, it seems to increase in the heart and the brain regions, and a slight increase in the heart. With the males, it doesn't seem to be as much accumulation going on in the brain, perhaps in the hypothalamus, but there is also an accumulation going on in the heart, kind of a similar pattern to the females. We also looked at iron because we were concerned about hexavalent chromium being reduced to trivalent, and then that trivalent chromium competing with trivalent iron for positions in the red blood vessels. So after 24 hours, what we saw with the females is sort of this trend for decreasing iron levels in the brain and in the heart, whereas with the males, it wasn't it wasn't really an effect. But 90 days, again with the females, we see this trend for increasing iron levels in all brain regions and also in the heart. But again, with the males, not so much going on in the brain, except for maybe this hypothalamus. In some of these preliminary results, we have some evidence that suggests there's increased neurodegeneration going on in the cerebellum, the cortex, and the hippocampus, and I'm currently working on scoring these samples blinded. We have evidence that suggests gamma-H2X expression is increased in the brain regions, with the foci formation is increasing, and we have brightly labeled nuclei, which, as you can see in those images, is a noticeable difference between control and treat. And these I'm also starting to score blinded. We have suggestive evidence for chromium accumulation in the left ventricle of the heart, especially after 90 days. There's like I said, up to a 17-fold increase in chromium accumulation in the heart. And we don't know a whole lot about how chromium affects the heart. And we need to clarify evidence for chromium deposition in the brain after the 24 or 90-day or pharyngeal exposure. There's a few different reasons why these results may seem a little weak. One is there's possible issues with chromium being cleared from the brain. As I mentioned, these animals were treated once per week. And one thing that's been studied more so in the last 5 to 10 years is something called the glymphatic system of the brain, which is kind of like the lymphatic system of the body, but it's focused on clearing stuff out of the brain, and it kind of runs along underneath or alongside all the cerebrovasculature. There are also some technical shortcomings where we only had 3 or 4 animals per group. So it's a small sample, and also the tissue is really small. As I understand it, it's ideal for ICP-MS to have 1 gram of tissue or 1 gram of sample to run. And I think for the cortex, which was our biggest sample, the average was maybe 0.8 grams. Whereas the hypothalamus, which was the smallest, was less than 0.2 grams. So it's really small samples that we're working with. There's also the chance that chromium is concentrating in subregions of these tissues, such as with the hippocampus, it could be in the CA1, CA2, or CA3 region, rather than the entire hippocampus. And that would require a much more robust and different approach to quantify, other than ICP-MS or AAS. So I'd like to conclude with acknowledgements, thanking all the members of Luke Hai's lab and the Wise lab that have participated in this animal study, particularly Dr. Luke Hai for all of his guidance over the last year and a half to get to this point. I'd like to acknowledge Amy Kai, who supported us in creating some of the artwork for our figures. And my funding, I'm on the T32 under Dr. Hein's grant. The animal study was paid for by Dr. Wise and Dr. Wise's R01, and Chris States' P30 is supporting this program. And with that, I'll take any questions.
Thank you, guys. That was talk. Now, we'll take any questions from the audience.
Hi, this is Bert Watson. This is a really exciting project. I'm looking forward to seeing what else you find as you go along. So I was caught by your route of administration. I'm reminded that the olfactory nerve is an important delivery route. Like I remember hearing that with manganese from welding fumes. It's a way to get into the brain, cross the blood-brain barrier. Is that part of the rationale behind using this oropharyngeal administration?
So the primary goal for the animal study was to look at lung toxicity. And the first time Wise Lab approached this, they did an intranasal exposure. And when they looked in the lung, they couldn't find any chromium. So they switched to this oropharyngeal to try and study chromium in the lungs a little bit better. And that's why we're using this inhalation approach. But yeah, the olfactory bulb is of course very important as an exposure route, because that also directly bypasses the blood-brain barrier. And the one challenge with rodents as a model for intranasal is they have much longer nasal turbinates, the air passages through their nose, and a lot of times stuff can just get caught in their nose and not actually reach their lungs or their brain. So with the oropharyngeal aspiration, we were hoping to get around that, get the chromium into the body. Whether or not the olfactory bulb is the primary route or it's via inhalation and sort of delivery by the blood, it's unknown for chromium in the brain at the moment. But it's hopefully something we can start to answer.
John, this is Greg Barnes here. I just wanted to ask a question. I was struck by your metals data in the brain. Is there similar data for chromium in the brain in humans as well, or differences in symptoms that are sex differences?
I don't think there's anything for sex differences with chromium in the brain, but certainly chromium levels have been measured in a variety of brain regions. I did show one graphic that kind of summarized that, with the pituitary having the highest levels reported. Those studies have varied from just kind of looking at element composition of the brain in infants and kind of like every 10-year age group. And one of the things that one of those studies found was that if you group all the adults together, there's a significant increase in chromium in that group versus infants and children. There's only a handful of studies that have looked at chromium in the brain in humans. I think that's largely because there hasn't been a significant discussion in the field about how chromium might be neurotoxic, which is what I'm hoping to start.
Christine, I see a couple of hands up, so if you can.
Oh, hi Yen, do you have a question?
Oh, yes. Thank you, Johnny. It's so excellent presentation. I have one question. So you have seen the chromium cause DNA double-strand breaks in the rat brain. As we know, cells has DNA repair pathway to try to repair these DNA double-strand breaks. So my question is, which DNA repair pathway will be disturbed? So in your hypothesis.
Right. Well, in part that will depend on which cell type is affected. For example, with neurons, they are postmitotic, so they do not enter the cell cycle, and thus they do not really employ homologous recombination for DNA double-strand breaks, which leaves NHEJ, which is a quicker, dirtier version of repair for neurons. But for the glia, like the astrocytes or the microglia, that could be HR or it could be NHEJ. It's a kind of a loaded question to hand because, like I said, like I'm saying, it depends on which cell types are affected. And chromium is also known to impair those DNA damage repair pathways. So that could contribute to more of the genotoxic problem occurring in the brain. But the exciting part is that we're seeing this evidence of DNA damage in the brain, even though we're not really seeing chromium levels increased in the brain. Clearly, it's doing damage regardless of whether or not it accumulates.
And Dr. Chin, do you have a question? Okay. Dr. Gazal, do you have a question? You have your hand up.
Yes, I do. I have a question regarding what do you think the mechanism is? There is very old literature on chromium, oxidative stress. Paul Oxteen, Alex Sivanian from USC, and Joe Randolph, if I'm not wrong, they published a lot on that. Do you think this is the mechanism? And my second question is, many people are taking chromium picolinate as a supplement to improve their brain function. There is also an old paper that said it improves the stroke outcome in a rat model. I think I read that a long time ago. What do you think about it?
Well, as far as the chromium picolinate, that's a trivalent form, so that's less toxic.
Less toxic or beneficial?
Well, the current argument for, as I understand it, is that sort of the lead argument in the field is that chromium is not an essential element. There is evidence in the literature that, like I pointed out, in the pituitary, which is kind of the area that's associated with hormonal regulation, which is what a lot of the chromium picolinate is targeting, the chromium seems to be 10 to 100-fold times higher than other brain regions. On the one hand, that might just be because it's got a weaker blood-brain barrier, because it's in that hormonal axis, or it could be that there is a regional essential role for chromium in that part of the brain. It would be a very interesting thing to look at. And on another point I'd like to bring up is Parkinson's disease patients. There's a couple studies that have measured chromium in the cerebrospinal fluid and have shown that there's about a 20% decrease in chromium in the cerebrospinal fluid of Parkinson's patients. I don't know what that means, but it could implicate, maybe an essential role.
Well, the next step is the mechanism, right?
Yeah, I mean, so as far as chromium picolinate being beneficial to the brain, I think for a healthy individual, it's not going to have any effect. There's potential it might have a pharmacological effect for diabetics or maybe for Parkinson's patients. I'm not sure. That's something that would certainly be interesting to delve into a bit more. As far as mechanism, yeah, chromium is known for inducing ROS, and a lot of literature has looked at lipid peroxidation. Most animal studies have reported some kind of lipid peroxidation a few have reported reaction to oxygen species, but with very limited quantification and analysis.
There's old literature. It's Dr. Augustine and Sevanian at University of Southern California and Joe Randolph, but it's extensive.
Yeah, and especially given that oxidative stress is an important etiological factor for a lot of neurodegenerative diseases like Alzheimer's and Parkinson's and ALS. Those all are featured with heavy oxidative stress and oxidative damage. It adds to the question of, is hexamethylchromium gerontogenic? Is it neurotoxic? And especially given that oxidative damage pathway, I think that's going to be an important role. Now, personally, I'm more interested in the genotoxic effect. On the one hand, because it's well established for breaking DNA, I've done my own research on that as I've grown as a scientist, but also the concept of genotoxicity in the brain is largely untouched. And that's really exciting to me, especially, when you look at
That's very interesting.
especially when you look at all the other cells that are involved with maintaining the brain health, and those are all mitotic. There's got to be some kind of genotoxic effect with those cells at least. Yeah, thank you for your questions.
Okay, Dr. States, do you have a question?
Yeah, some nice preliminary data, Johnny.
Thank you.
I was struck by the difference in toxicity of potassium and sodium chromates. And I think I missed your explanation for the difference.
So on one hand, it could be due to uptake, and that's the next thing I'll be looking at. It could just simply be that potassium chromate gets into the cells more easily than sodium chromate for this cell line. Or it could be that sodium somehow benefits the cell line that in a way that potassium doesn't. It's hard to say at the moment. It's just a very intriguing result.
Could it be the potassium itself? I know the concentrations are pretty low, but
You mean potassium causing toxicity?
I'm just wondering.
I would doubt that, because potassium and sodium are essential elements for cells, and yeah, again, potassium is pretty low in this case. Chromium is the much more toxic chemical there between those.
Yeah.
I am also looking at zinc chromate, but that's a particulate, so that it's not as readily comparable until I do the uptake for that as well. And I'll have the summary of those results probably this weekend.
So in regards to which cells in the brain are actually getting hit, can you do the histology on those slides you have for your gamma H2AX? Can you identify the cells?
Yeah, and that's what I plan on doing. Is first I want to figure out which regions are affected, and that's why I'm just looking at the nuclei and gamma H2AX. Then the next layer will be to add a marker for neurons, a marker for astrocytes, a marker for microglia, and try and hone in on which cells in those regions are most affected. Because that will change the questions that follow immensely.
Yeah, that's also critical support for the hypothesis as to which pathways you're going to look at.
Yeah.
Okay, thanks.
Yeah, thanks for the questions.
And Dr. Hong, do you have a question?
Hi, Johnny. Very fascinating project. Just quickly, I think it's puzzling that there was a little bit of discrepancy between your Comet assay and the gamma H2AX results, because gamma H2AX, there's a robust increase in response to chromium, but not so much with the Comet assay. I mean, it could be due to the sensitivity of the assay and stuff, but I remember that it could be due to, so gamma H2AX is mostly induced by DNA double-strand breaks, but sometimes it can be induced by apoptosis too. So if the chromium, instead of causing DNA double-strand break, but causing apoptosis, I wonder if that could be, that could explain your that discrepancy. And my second comment is that I don't know too much about this, but I know that there's some neuroinflammation is now kind of lot of people are interested in that in terms of understanding the neurodegenerative diseases and stuff. So I was wondering if you're planning to look at any type of inflammatory markers or cytokines in chromium treated brains.
Yeah, so for the Comet assay, no, like I mentioned, one idea could be that if one cell is being affected of the 8 or so that are there, that effect's going to be just totally washed out. And that's why we don't see anything with the Comet assay. The other thing was, this was our first attempt at doing this with tissue. Like I've said, the WISE lab is very proficient at doing this with cell culture, but we haven't optimized the Comet assay technique for brain tissue in particular. And so perhaps, the controls are during the gel electrophoresis are being brought out. So as far as the treated animals or the damaged cells, and rather than that being broken DNA, it's just the DNA is being unwound by not having the assay optimized. As far as neuroinflammation goes, yeah, that's something I'm definitely planning on looking at. That's kind of the third endpoint. So first I'm doing the DNA damage, then I'll figure out which cell type and look at cytotox versus senescence, and then I'll look at inflammation. And as you mentioned, some of the H2X could be due to apoptosis, and given that I am seeing neurons are dying with that silver stain, that could simply be the explanation. And one way I can probe that is using caspase-3 with staining and seeing if there's increased caspase-3 expression in the cells that have increased gamma H2X. That's certainly another question for the future to be addressed as far as teasing out what's exactly going on here. And for the neuroinflammation aspect, two things I can focus on histologically are the morphology of microglia and the morphology and relative expression of some markers in the astrocytes. And those are pretty common techniques for looking at neuroinflammation and histologically. I've also got tissues that were harvested for protein and RNA analysis. So with those, I could do some ELISA kits to look at some of the cytokines important for neuroinflammation. And is also something I'm very much planning on doing, but probably won't get to that part until the summer or the fall after I've gotten these other couple endpoints worked out.
Great, thank you.
All right, any other questions? Okay, thank you, Dr. Wise. That was a great talk, and thank you all for joining today.
Thank you.